Hello, my name is Dr. Cara Cargo-Froom, and today I'm going to be discussing capturing equine digestion in an in vitro protocol. So what is in vitro? I think that's the first important question we need to ask about today's talk. In vitro is the simulation of what occurs in a living organism, whether that's an animal or a microorganism, in a lab setting. So what we're trying to do is we're trying to apply physiological properties from an animal in an in-lab setting, absent of any animal use. So a great example of in vitro that a lot of people experience every single day is in vitro fertilization. So in vitro doesn't just apply to digestion. It can apply to many different areas of animal use or animal studies because we as humans are animals. So if we think about reproduction, we can do that naturally, or we can take it and flip it into a lab setting. The same goes for digestion. So what in vitro digestion is trying to do is it's trying to simulate that in vivo digestion in a lab setting. So that digestion that's occurring in that live organism in a lab setting. Why do we want to study digestibility and fermentability? I think that's one of the big questions we need to address for this topic. And one of the reasons is because compared to other animal species, such as livestock production species, like our cattle, swine, and poultry, there's very minimal understanding on the digestibility of even our most common feedstuff in horses. While there is some data out there, it's not nearly as robust as others. So what digestibility and fermentability values can give us is the amount of nutrient that's actually available to the animal for absorption. When we don't have these values, we only know what we're offering to the animal via the feed composition. So we know how much of a protein, fat, mineral composition exists in that ingredient, but those nutrients are not always available to the animal for absorption. Additionally, digestibility and fermentability provide us information on the efficiency of a feed, so how much that animal can actually turn that food or ingredient into an output. So that could be milk production, potentially it could be performance related, or maybe it's in relation to gestation and lactation. These values can also provide insight on the quality of a feed. Generally, a more digestible or fermentable feed is going to be higher in quality because it can provide more nutrients to the animal than a feed that is lower in digestibility or fermentability, which means we're going to need to feed more of that ingredient for the animal to get the same amount of nutrients compared to that highly digestible or fermentable ingredient. If feed X only provides 50% digestible protein and feed Y provides 80% digestible protein, that means that if you're feeding the same amount, feed Y is going to provide more protein to the animal. To provide the same amount of protein, that means you would need to almost double the amount of feed X that you're feeding to get the same amount of protein as feed Y. And so this is why digestibility and fermentability values can provide insight on the quality of your ingredient. So I've thrown the word digestibility and fermentability around now quite a bit. But what is digestibility and what is fermentability? They are different because digestibility refers to the breakdown of food by the animal themselves. So this is from digestive juices as well as enzymes that the animal secretes to help break down their food into those smaller subunits for absorption. When we compare that to fermentability, it's a little bit different because there are certain nutrients such as fiber in foods that an animal cannot digest themselves. We simply do not have the secretions, the digestive enzymes, to break these nutrients down. Instead, the microbiome of the hindgut can extensively ferment this ingredient. And through fermentation, we end up with a release of other nutrients such as volatile fatty acids and B vitamins, which the horse can then utilize. So while the end result is the same, that the horse can absorb these nutrients. What is different is how the nutrients are released from the ingredient. Digestibility refers to the animal's ability to break down that food, where fermentability refers to the microbiome that exists in that animal to transform the food into nutrients that the horse can utilize. So before we start talking about how we can measure digestibility and fermentability, let's discuss the benefits of these values to the horse industry. As nutritionists and horse owners, we can greatly benefit from having digestibility and fermentability values in literature because this will help more accurately meet the needs of the horse's nutrient requirement. All animals have requirements for certain nutrients. However, not all foods are going to deliver the same amount of nutrients or even have the same level of digestibility and fermentability. By having these values, we can then incorporate digestibility and fermentability coefficients into our formulation for diets, which means we're better capturing how much of a nutrient will then be available for the animal to absorb. Through improved rationing, we can also reduce the risk of disease. Now, this can be either through a reduction in overfeeding, which could reduce the possibility of obesity, or by better meeting the nutrient demands of the animal. Thus, we're not overfeeding energy that could result in obesity, but we're also not underfeeding so that there isn't sufficient energy or micronutrients for proper physiological processes. Additionally, we can better manage diets of horses that may be ill or diseased. A great example is by determining digestibility and fermentability, we can also determine the rate at which certain nutrients are degraded, so digested or fermented, and how fast those nutrients are released. So this could be especially important for horses that may be prone to colic or where laminitis is a concern, and we want to control sugars, etc. By more precisely feeding an animal to their nutrient requirements, we can also reduce our environmental footprint. When we overfeed a nutrient to an animal and we exceed their requirement, those excess nutrients aren't going to be utilized, and instead they're excreted as waste, whether that's fecal waste or urinary waste. And in the case of our equine species, our livestock species, really any species in general, this means we're contributing additional nutrient waste to the environment. And while for one animal, that may not be a large environmental footprint, when we consider the waste of many animals, it can add up over time. So by better feeding and meeting those specific nutrient requirements, this means that there's going to be less excess nutrient waste in the feces and urine of the horse. And finally, through measuring digestibility and fermentability, we can also gain insight on how feed processing and storage affects nutrient composition of the feedstuff itself. Processing can negatively or positively impact changes in nutrient composition of a feed. The same can be said for storage. For example, some nutrients may degrade in quality over time, which means by the time you're feeding that feed at the end of its storage, it's not going to have the same composition as at the beginning. So by taking those parameters into account when we measure digestibility and/or fermentability, we can also figure out how those different factors influence digestibility and fermentability and potentially correct for or adjust for in our formulation for those specific parameters. However, there are some inherent difficulties that arise when we try to measure digestibility and fermentability in an animal. So using animals as our digestion models. One of the big concerns is animal welfare. So there are 3 R's in research that we need to consider when utilizing animals. These are replacement, reduction, and refinement. Replacement means, can we replace this animal through another model, i.e. in vitro? Reduction is, can we reduce the number of animals utilized overall? And refinement is, can we refine our process to either eliminate invasive and potentially painful procedures on the animal Or can we refine it so that we need less animals to do said procedures? Animal welfare is a really large concern in our current world. We want animals to be treated the best way possible. So in addition to that, there's been more restrictions that have been put on animal use in research over the past couple decades. We need to go through processes to have animal utilization approved by an animal care committee, and they have oversight on what procedures we are allowed to and not allowed to perform on animals. And so by replacing animals with an in vitro, so an in-lab system, we remove this animal welfare concern. There's also reduced control when we're using an animal model versus an in-lab model. And this is because in lab, we can closely control all of the different parameters of our study, whether that's the concentration or how much of a solution we're adding in, the specific activity of enzymes, even how long a food sits in the digestion chamber. But this isn't always controllable in animals. We cannot always control the gastrointestinal transit time of an animal, the enzyme activity that's occurring, the hormone signaling and feedback. And there are also other variables that could affect it. Perhaps an animal becomes sick and needs to be removed from study. Sometimes we can't get animals to eat, and if we can't get the animals to eat, then we can't get the measurements we need because digestion is all about feeding. It's also much more challenging to measure digestion in an animal compared to an in-lab setting. And a great example is there are many different approaches we can actually use to measure digestion in an animal. We can use total tract, which measures the ingredient that's been eaten and the final output in feces. But we can also measure ileal digestibility, which is the ingredient from when it's being eaten, and it's sampled before it enters the hindgut, so enters into the cecum and colon. So it's actually sampled at the end of the small intestine. But this method is quite invasive because we need to surgically insert a cannula, so a tube that leads from the small intestine to the outside of the body so that we can take these samples. Well, that involves animal surgery. So right there, we tie back to our welfare concern. It's also more complex and more costly. So we often revert to methods such as total tract digestion, which is less invasive, but there are inherent variations and physiological influences that can actually lead to a less accurate measurement of digestion because of how digestion occurs and fermentation versus digestion. And finally, animal studies are often much longer than our in-lab studies. And this is because when we're feeding an animal, we actually have to allow their digestive tract to adapt to the new diet before we take any samples. And this is because diet composition can influence how much enzymes are secreted into the small intestine to digest the food. It can also change the microbiome of the hindgut itself. So if there's a lot of fiber in the diet, the microbiome is going to need to adjust because they now need to adjust to higher fiber intake. If fiber is reduced, again, that microbiome is going to adjust because there's less substrate to feed those microorganisms. So we need to allow usually at least 5 to 7 days for dietary adaptation before we even start to think about taking samples such as fecal samples or ileal samples. And sampling usually occurs over a period of 2 to 3 days. So all in all, you're looking at a week to a week and a half to evaluate one diet in an in-animal setting. When we're in lab, we can do this quite rapidly. We don't need to allow for dietary adaptation. Beakers don't need to adapt to the diets. We can readily feed the substrate in, add in all of our solutions, digestive enzymes, and begin digestion straight away. We can also sample straight away. We don't need to do multiple days of sampling to ensure that we have a representative sample. So while an in-animal study may take a week to a week and a half to generate data for one ingredient or one diet, we can generate the same information in an in vitro, so an in-lab setting, within a couple days. So I think it's important, before we start to talk about how we replicate digestion in the lab, to talk about what is digestion in the horse? How does digestion of food occur? Because every single animal digests food as nutrients, but there are different steps along the way that occur so that these nutrients can be released or produced for absorption. What a lot of people don't realize is that the first step of digestion actually starts in the mouth, so when the food enters the digestive tract. What's really cool is the digestive tract is actually considered an open system, so it's not actually part of your body, so it's not considered inside the body because it's an open hole from start to end. It's one long big tract. And in the mouth, teeth act to break down food into smaller particles. The smaller the particle, the more easily accessed for digestion. So this allows enzymes to function more optimally because there's greater surface area for digestive juices and enzymes to access the food and release nutrients. In addition, saliva is excreted in the mouth. Unlike many other animal species, there's very little starch digestion that occurs in the mouth of horses. They have much lower amylase activity compared to, say, humans and pigs, etc. So really what saliva acts as in the horse is a buffer. It mixes with the food and it allows for food to be easily swallowed. Once swallowing occurs, the food is transported from the mouth down the esophagus to the stomach. And this is actually through movement called peristalsis, which occurs throughout the entire gastrointestinal tract. And this is involuntary muscle movements that help move your food along as it's digested in the different compartments. So once food moves from the mouth down the esophagus, it enters the stomach. And in the stomach, there are two specific secretions that occur. This is stomach acid, so hydrochloric acid, and pepsin, which is an enzyme. Acid works to continue to break down the food into smaller particles, while pepsin starts the process of protein digestion. What's interesting is when food starts to mix with acid, you'll actually see a drop in pH in the stomach. And this is because the stomach environment and pepsin works best at an acidic pH, so a pH generally between 1 to 2. Once food has finished being digested in the stomach, and this can be anywhere from 15 minutes to 2 hours, depending on the amount of food fed as well as the food type fed, food then enters the small intestine. And the small intestine is where the major site of nutrient digestion and absorption occurs. Now remember, digestion is different than fermentation. So fermentation is going to occur in a different part of the digestive tract. But this is where a lot of our proteins, our carbohydrates, and our fats are broken down into smaller subunits for absorption. We also have a major site of mineral and vitamin absorption in the small intestine as well. And in the intestine, different digestive juices and secretions, as well as enzymes, secreted into the gastrointestinal tract to continue breaking down food. So let's talk a little bit about the different secretions that occur in the small intestine and why they are important. The first secretion that occurs at the very beginning of the small intestine as food moves from the stomach into the intestine is bicarbonate. And this is because, unlike in the stomach, the enzymes that are secreted in the small intestine function better at a higher pH, a more neutral pH. So bicarbonate, which is a basic solution, is secreted into the intestine to balance out that acid and increase the pH anywhere from about 6.3 to 7.6, depending on the different segments of the gastrointestinal tract. And again, bicarbonate is needed because not only do we need to have a more basic environment for the enzymes to function, but we also want to ensure that the lining of the gastrointestinal tract isn't damaged by acid. So it helps buffer the lining so that we can have optimal absorption of nutrients. Another major secretion in the gastrointestinal tract is enzymes. So while there are some enzymes, like pepsin, that's secreted in the stomach, most enzyme activity is going to occur in the small intestine of the horse. And there are different enzymes that are secreted. So there are different enzyme groups that act upon different nutrients or different categories of nutrients within the body. So our first group is amylase. And amylases work to break down our carbohydrates, so our starches, not fibers, because fiber is a carb, but it's not a digestible carb like our starches are. And it breaks down starch into smaller subunits, so glucose, fructose, and galactose, for absorption that's then utilized for energy. I think a great example of how enzymes work and why they're so important is lactose intolerance in humans. I think most of us know someone who is lactose intolerant, and if you don't, you can easily Google what lactose intolerance is and the different symptoms that occur. The biggest symptom is gastrointestinal upset when someone who is lactose intolerant consumes too much lactose. Lactose is a carbohydrate, and it's made up of two subunits, galactose and glucose. And these are the absorbable subunits. But lactose itself cannot be absorbed from the small intestine. So lactose needs to be digested by amylases before it can be absorbed. However, in people who are lactose intolerant, they either don't produce enough enzymes or they don't produce the enzyme at all to be able to digest lactose. So instead, lactose remains intact in the gastrointestinal tract and it cannot be absorbed. And this is what causes digestive upset. And this is why enzymes are so essential to digestion, because without them, we could not break our food down into the small absorbable subunits. Other group of enzymes that act in the small intestine include lipases, which break down fats, and proteases, which break down proteins. And through the action of these enzymes, we can then absorb nutrients to transform into other compounds or use for energy within the body. Another secretion that's really important in the digestive tract is bile. Bile is secreted by the liver into the small intestine, and it's essential for fat digestion as well as absorption. Without bile, fat would not be effectively digested or absorbed from the gastrointestinal tract. Fun fact, horses, unlike many animal species such as humans, dogs, pigs, do not have a gallbladder, which means they can't store bile. Rather, bile is continuously trickled in from the liver into the intestine. And this is why, if you want to feed a higher fat diet to your horse, it is possible, but it's always suggested that you allow for a longer period of dietary adaptation and switch over to that higher fat content, because bile is not stored and the horse needs to adapt to that higher fat intake. Where in animals that have gallbladders, we can bolus, we can add more bile into the small intestine to deal with that higher fat intake. Finally, one of the last secretions that occur in the gastrointestinal tract, which a lot of people don't realize, is actually hormones. So hormones are an important part of digestion, and this is because they act as feedback and signaling loops. Some hormones can signal the release of certain enzymes, where other hormones may signal satiety, so the need to stop eating because you are full. And something I want to keep in mind as we then enter our in vitro, so our in-lab discussion, is that while other digestive secretions are often utilized in vitro, hormones are not. And this is because we don't have the ability to add in that hormone signaling or feedback loop in an in-lab setting, because it's not a live animal. It doesn't need signals to the brain to then tell the animal what to do, what to stop, what to eat, etc. Once food has been extensively digested in the small intestine, it then moves into the hindgut. So we'll refer to the hindgut as the cecum and the colon together. The first step in the hindgut is that food enters the cecum, so digesta enters the cecum. And in the cecum, it is extensively fermented by the microbiome. So the microbiome consists of yeast, bacteria, protozoas, these small living organisms that we can't see, these microorganisms that can digest fibers that the animal is not able to digest themselves. In the cecum, that food substrate that's left over, that isn't absorbed from the small intestine, is extensively fermented by the microbiome, and nutrients such as volatile fatty acids and B vitamins are produced so that the animal can absorb and utilize these nutrients. Our digesta then moves into the colon from the cecum, where fermentation continues to occur. In the colon, we also have mineral and water absorption that occurs, but this absorption isn't to the same extent as absorption from the small intestine. And at the end of the digestive tract, any nutrients and or other metabolic waste, bacterial waste, etc., is excreted as feces. So the undigested food moves into the rectum where it collects until there's sufficient waste to be excreted, and then is then excreted into the environment. And if we think back to previous slides and previous discussion, this is where we see environmental impact if we're overfeeding nutrients that might not be absorbed and then are then excreted back into the environment, building up that nutrient concentration either in soil, water, etc. Now that we've reviewed how digestion occurs in the horse, let's look at how we try to replicate digestion in an in vitro system. So this means in lab. This is a slide that has a lot of information, but I think is helpful because there are so many different approaches that can be taken to digest an ingredient in an in vitro system. We have static digestion, which can include pH drop, pH stat. It can also include two-step digestion methods such as pepsin pancreatin and our two-step digestion, and hindgut fermentation methodologies. And these are all considered static digestion methods, which is what we're going to be addressing in today's discussion. But there are also dynamic methods that we can use. And the reason why I want to point these out is because dynamic methods more closely try to simulate the digestive tract. And this is because in static methods, we don't get the same actions such as peristalsis, digesta mixing, pH control, enzyme secretion, as we do in a living animal. So these dynamic methods are almost a bridge between static and in vivo systems. It tries to better replicate peristalsis, and it uses actually a computer model to determine when certain enzymes should be secreted, when food should move from one compartment to another, and when acid-base balance needs to be changed. So there's a little bit more control in our dynamic systems than our static systems. However, with our dynamic systems becomes more complexity as well as an additional cost. And in certain studies, especially human in vitro studies compared to in vivo studies done in rats, there's a relatively high correlation between these two values for static methods. So while dynamic methods try to best simulate in vivo digestion, static methods still have their place and are simpler to implement and are often more times cost effective. For the purpose of today, because there are so many different methodologies that are currently existing for in vitro digestion, we're going to focus on our static methods. And we're going to try and tie in those different steps to those in vivo steps, so those in-animal steps of digestion in the horse. The first method we'll go over is pH drop digestion. pH drop is one of the simpler in vitro methods to implement, and this is because it isn't trying to replicate the different digestive compartments such as the stomach and small intestine, hindgut of the horse. But rather, we're looking at the change in pH of a solution over time. So starting from a basic solution, so a higher pH, and dropping down to a lower pH. The premise of this approach is that we start with our food substrate and a solution that is at a high pH, so it's at a pH of 8. We then add in a specific enzyme mix to start digestion of the food substrate. After allowing this mixture to sit for a set period of time, usually 10 minutes, We then start to measure the drop in pH as those nutrients, as that food is digested and those nutrients are released. By plotting the change in pH over time, we can then use regression equations to calculate digestibility. So we aren't actually measuring the nutrient intake versus nutrient output, but rather we're using these graphs that are produced and then the lines that are produced within these graphs to create equations to determine digestibility. pH stat digestion is similar to pH drop, as it's not replicating the different digestive compartments of the animal. However, the difference is, instead of monitoring the drop in pH over time, we're looking at the maintenance of pH. So we want the pH to stay the same. This is achieved through mixing of a solution and the ingredient substrate that we're interested in, and bringing the pH of that solution up to 8. An enzyme solution is then added in to start digestion. But rather than watching a drop of pH over time, what we're doing is we're then titrating in a solution to ensure that the pH stays at 8. So rather than looking at a drop in pH over time, our graph is going to give us how much of that solution was consumed during the digestion process to maintain our pH at a level 8. Again, this is plotted on a graph, and then we use that graph and line within the graph to create regression equations so that we can determine digestibility. However, there are also in vitro systems that try to replicate the different digestive compartments of an animal. One such step is the pepsin-pancreatin method. So this aims to mimic the gastric compartment as well as the intestinal compartments of the horse, so the stomach and the small intestine. Fermentation is measured separately, but these can be done in successive steps if you want to look at digestion from start to finish. The first start of the pepsin-pancreatin digestion is the gastric phase. This is where hydrochloric acid and pepsin are mixed with your ingredient substrate to start digestion. And this is where protein digestion will begin to occur. The second step is the intestinal phase, and we can break this down into 2 sub-steps. The first is that we need to neutralize that acidic starting solution for the gastric phase. So we need to bring the pH up similarly to what bicarbonate would do in the small intestine. So we often add in a basic solution such as sodium hydroxide to increase the pH. The second step is then to add the pancreatin, which is an enzyme cocktail, to our substrate so that we can continue enzymatic digestion of our ingredient of interest. So what pancreatin contains is it's often a mix of amylase, lipase, and protease to break down our food into those smaller subunits that would be absorbable by the animal. Finally, we have determination of digestibility. So once we have finished those specific digestion steps, we take that substrate and we analyze it. And we, through different mathematical equations, can input that data as well as the starting nutrient concentration of the ingredient before it was digested and determine digestibility through calculations. Similarly to the pepsin-pancreatin method, the 2-step digestion method aims to simulate the gastric and small intestinal phase of the horse. However, there may be some differences from the pepsin-pancreatin method in the 2-step digestion method. So these differences could be a variation in the solutions and concentration of the solutions used, the enzyme cocktails used, as well as the time that the digestion is incubated for. So time may change. Just like we can represent gastric and intestinal digestion of nutrients, we can also represent that hindgut fermentation, so the fermentation that's occurring in the cecum and colon of the horse in lab. Now this is a bit of a different approach compared to our other methods we discussed because these don't use enzymes at all. Rather, samples from the cecum or from feces, or a combination of both are taken. And we can isolate specific bacteria and microorganisms from those samples into what we call an inoculum. So that inoculum is what we're going to use to create that fermentation process of different ingredient substrates. So the first step in hindgut fermentation is taking that inoculum and adding it to your substrate. Then we allow that substrate and inoculum to mix and sit, usually for a period of about 48 hours. And while that substrate is sitting and fermenting, we're actually measuring the gas in a closed system over the period of time. So how much gas is produced? Because gas is one of the different products that is produced during the fermentation process. So in addition to vitamins and volatile fatty acids, we also have gas production. And through measuring this gas production, we can then calculate fermentability based off of, you can guess it, regression equations. So we talked about regression equations for pH drop, pH stat, but we can also apply regression equations to determine fermentability. Now I'd like to address some of the advantages and limitations that come along with static methods. Because while there are benefits to utilizing these methods, there's also only so much of digestion we can capture in them. One of the first advantages of in vitro digestion is that we have no animal welfare concerns, which means we have no ethical concerns about using this approach. This means that we don't have to get animal approval usage, and that the studies can begin as soon as you have the correct equipment, the correct solutions, and your feed ingredients. It's also extremely reproducible. So what this means is within the same lab running a study, or across different labs running studies, you'll often yield the same results. And this is good. We want a reproducible method that yields the same results, because this means that as long as you follow the method, you're going to have the same outcomes. If the methods didn't provide the same outcomes each time, we wouldn't know how reliable these methods are for determining digestibility and fermentability. We also talked about control earlier. So one of the great aspects about in lab setting is the amount of control we can influence over a study or procedure. We can control every single aspect of what's occurring, make detailed notes so we know exactly where something may have gone wrong, why something may or may not have digested appropriately, where things may need to be changed. And through this level of control, it allows us to really understand the influence of the different parameters over our eventual outcomes. Another advantage of in vitro systems is that they are rapid and relatively simple. It's quite easy to teach someone how to implement these methods as long as you have the appropriate equipment and chemicals available. It's also much quicker compared to our in vivo, so our in-animal trials. And this is because, like we've mentioned before, you need to allow for dietary adaptation of ingredients. Sampling often takes longer, and then the analysis may even take longer, whether that's due to drying and grinding of the samples taken, etc. In an in-lab setting, most digestions are done within a couple hours to a couple days, rather than multiple days or even weeks, like we see in our animal studies. And finally, in vitro is also quite cost effective. So while there may be a cost associated with either paying someone to analyze the samples or the equipment, solution, etc. that you need, there's no additional associated cost with feeding, housing, and caring for the animals, which is all something that needs to be considered in an animal study, especially because we never run a study with just one animal. Generally, there's going to be multiple animals, and in large industry settings for research, that can get up to even hundreds of animals. However, there are some inherent limitations that come along with an in vitro system. And one of the biggest limitations is we can't capture that dynamic, ever-changing nature of digestion in an in vitro system. When we think about digestion, it's constantly changing depending on what's been consumed, how much is being consumed. That can influence gut transit time. It can also influence intestinal secretions. It may influence enzyme activity. For example, if you have a diet that's quite high in fat and protein, but low in starch, there's going to be little amylase activity occurring, but much higher protease and lipase activity occurring. Where in an in vitro system, we need to standardize this more. So you're going to select specific enzyme activities for your digestions that may not always correlate to the nutrient content of the ingredient that's being digested. It's also much harder to capture gut transit time in an in vitro system, especially for our static systems. We generally select a specific incubation time period. However, as we know, just like in humans, in animals, gut transit time can vary tremendously. If too fast of a gut transit time, perhaps something upset the gastrointestinal tract, or there was excessive fiber in a food, food may move through that gastrointestinal tract faster. Perhaps an animal ate a large meal, and that meal is sitting in that gastrointestinal tract longer. That might actually increase gut transit time, and so that food remains in the gut longer, which means there's more chance for absorption of nutrients to occur. In addition to capturing the dynamic nature, static systems also don't capture nutrient absorption. So no nutrients are being absorbed from those beakers or containers that fermentation and digestion are occurring in. But in an animal, we have those disappearance of nutrients from the GI tract into the circulatory system to eventually be utilized. And that's something that we're missing in an in-lab setting. There are also no physiological feedback loops. So when we think back to the hormones that we discussed and how they can help regulate digestion, either through satiety signaling or through the release of enzymes, we don't have this feedback occurring in vitro. And these feedback systems are an important part of digestion and metabolism in an animal. And so to be lacking that specific feedback loop, we're missing a part of that overall digestion picture. And finally, while we do isolate inocula, we are missing the majority of the gut microbiome. So there are some microorganisms that exist in the small intestine and in the stomach, even in the mouth. Obviously, there's extensive microorganism activity in the hindgut, so the cecum and colon. And this is not represented in our in vitro systems. We're missing a large population of those microorganisms. So even though we can capture fermentation through inocula, we aren't capturing the same diverse, extensive microbiota that exists in an animal. So how do we ensure that in vivo can be reliably replicated in vitro? We try to aim to meet these 5 specific steps. The first step is the sequential use of digestive enzymes that occur in the animal. For example, we're not going to use an amylase or lipase in the stomach section because that isn't what occurs physiologically. Rather, we want to ensure pepsin is being used in the stomach portion because that is what occurs physiologically. We also want to make sure that our in vitro system is at the appropriate physiological stage. So that's temperature and pH, etc. So all of these little different parameters that occur within an animal. If the internal temperature of a horse is 39 degrees, then we shouldn't incubate a feed item at 36 or 37 degrees Celsius, as this may have influence on the overall digestion or fermentation process. So we should try and replicate that internal temperature to the highest degree possible. Similarly with pH, we want to try and replicate the gastric pH portions of the stomach and the small intestine to meet what would be occurring in the horse. We can change pH quite readily to any degree. However, if we aren't replicating those physiological parameters from the animal, how are we going to accurately develop coefficients for digestibility and fermentability? We also want to make sure there's appropriate digestion and enzyme mixing. In our gastrointestinal tract, food is constantly being swished around, moved back and forth through peristalsis. So we want to ensure this mixing is being replicated in an in vitro system. We also need to look at gut transit time. What is the average transit time for that feedstuff or ingredients in general, or just in general for the horse? We want to try and mimic that to the best degree possible. So if the average stomach transit time is approximately 1 hour, then we should consider digesting our gastric compartment of our in vitro system for approximately 1 hour. And finally, we should also consider the removal of digestion products. So are we able to remove some of that substrate during, especially as we move from compartment to compartment, for example, from small intestine to the colon or the cecum, more correctly? Can we capture that movement from the small intestine in vitro into our hindgut fermentability? This will try and somewhat replicate nutrient absorption, although we simply don't have the capacity to completely mimic nutrient absorption from the gut in a lab setting. So I've thrown a lot of science at you now, but let's talk about how this applies to the equine industry, so our equine sector. How can we fit in vitro characterization and digestion and fermentability of feedstuff into the equine sector? I think the first thing to address is digestion and fermentation, and really food degradation. So digestion and fermentation fall under food degradability. So how is a food broken down? One of the reasons degradability is of interest is because not only can it help us better formulate diets, but we can use this information as inputs for equine models. So Dr. Emily Leishman talked about developing an equine model for digestion, and this is where my project actually fits into the overall bigger picture. So one of the reasons feed degradability is so important is because there are many factors that can influence it. So if we back up a little bit, feed degradability can be influenced by the feed characteristics and composition of the ingredient you're offering to a horse. And if you back up further, there are so many different factors that can influence feed composition and characteristics. These factors include crop management, geographic origin of the crop or ingredient that you've fed. So did it originate in Canada? Did it come across the seas from Europe? What about harvest and storage? So we talked about how storage can influence nutrient composition. Well, clearly this can then influence feed degradability. Soil and nutrient composition can also influence the nutrient composition and characteristics of the ingredients, and that's because plants rely on soil to provide them nutrition so that they can grow. And finally, plant variety can also influence the characteristics and compositions. Well, that two plants may be in the same family or even of a similar species but a different variety, these plants can have drastically different characteristics and nutrient composition. A great example are pulses, so things like chickpeas, beans, lentils, legumes. Often within a category, such as chickpea, you can have two different varieties, say Kabuli and Desi chickpeas. But their nutrient composition can actually vary greatly from one another. One may be higher in protein or minerals. One may contain more soluble fiber. And so these are all factors we need to consider when considering food composition and characteristics and feed degradability. As we talked about earlier for those digestion fermentation coefficients, which are going to be your feed degradability values, those can either be used for direct application, whether that's feed formulation, or we can use them as inputs for computer models to help us determine further digestion values without having to actually invasively use an animal. So the overarching goal for this study is to support the development of a mechanistic model for the equine sector to advance the field of equine nutrition. However, my project is much more focused, and it's specifically focused on the in vitro evaluation of feedstuff to determine digestibility and fermentability values, as well as to measure overall feed degradation. So the specific goals for my project are to assess the existing in vitro protocols that exist for both equines and other animal species, and then to select the most appropriate approach based on the literature that I have summarized and compared to one another. After we've selected the most appropriate approach, we are then going to use that in vitro methodology to determine both digestibility, fermentability, and the level of feed degradation that occurs in the foregut, so the gastric and small intestine compartments, and in the hindgut. And results from this specific sub-goal are going to be what feeds in as inputs for the equine digestion model. So we talked about those 5 items for successful in vivo replication. But specifically for my study, what do I need to consider to be able to best replicate digestion that occurs in the horse? First, I need to consider the digestive physiology of the horse. So compared to other animal species, especially other monogastric animal species like dogs, humans, and pigs, there's reduced amylase activity that occurs in the small intestine. We also have the constant trickle of bile. And there may be other different physiological processes that I need to take into account, like gut transit time. Also need to consider the acid-base balance. So what is the ideal pH in the different intestinal and stomach compartments of the horse? Are we at the right pH to ensure digestion is best simulated in the lab setting? We also need to consider enzymes. What enzymes are we going to add in? What activity are those enzymes going to be at? When are they going to be added in? Those are all factors that need to be considered. And then finally, the gut microbiome. So how are we going to replicate that extensive microorganism colonization that occurs in the horse in an in-lab setting? And how can we best represent that gut microbiome? To achieve this, we can break down the project into sub-objectives. So the first objective that we're working on currently is assessing the literature out there for in vitro digestion for equine, as well as other animal species, and then selecting the best approach to simulate gastric and small intestinal digestion. Currently, we're leading towards an either 2-step approach or pepsin pancreatin, as we believe that will best capture the digestion that occurs in the stomach and small intestine, and we can more accurately make changes to represent the horse physiologically. Then, we're actually going to implement that in vitro study and take a whole bunch of common feed stuff that are fed to horses in North America and digest those ingredients to determine overall digestibility, but also to determine feed degradability. So how quickly that nutrient degrades, how fast are those nutrients released? Then we'll take a similar approach for the hindgut. So again, we will take the current literature that's out there, assess that literature for equines, compare it to other animal species that have done fermentation studies in lab, and then we are going to apply it in an in vitro system. So we're going to take the best method, whether that's a method that currently exists or a method we choose to modify, and we're going to try and represent the hindgut fermentation that occurs in the horse in lab, using the same feed ingredients that we used for the gastric and small intestinal digestion. And we're going to measure fermentation, and again, nutrient degradability as that nutrient ferments. So a lot of information has been presented today. Let's revisit why it's important and what we hope to achieve as outcomes from this project. Firstly, we can improve ration balancing by understanding how digestible and how fermentable an ingredient is. And this can lead to reduced environmental impact, better nutrient delivery for the animal, and can also take into account disease and illness. So whether that's better formulation for horses that are diseased and ill, or an overall reduction in potential disease, such as laminitis. And through using these values as inputs for a digestion model, we can also optimize performance. So if you're interested in how performance can be optimized, I'd highly suggest looking at Dr. Emily Leishman's video, which ties in nicely and explains why modeling is important. And you can also see how these inputs would feed into the overall big picture. So how do we achieve this overall project? Well, It starts with a diverse collaboration of people with different backgrounds. For example, I can bring a background in nutrition as well as in vitro digestion to the team. But we need more than just that. We have experts in the field of animal modeling. We also have experts in the field of computer science and engineering. But one of our most important experts are our industry partners who provide oversight. And these industry partners can help provide oversight on the project so that we're ensuring we're meeting the needs of the stakeholders. So you at home who are watching this, whether it's horse owners, horse trainers, those who are feeding horses, by working with industry partners, we can ensure that we're not only meeting the needs of research and the academic stream, but also within the industry so that you as stakeholders can take something from the data we're generating. I appreciate the time that you spent listening to me talk, and I hope that you've learned something interesting, whether it was how digestion works, how we can apply it into a lab setting, or simply how this applies to the equine industry sector. If you have any questions, please do not hesitate to reach out, as we're always happy to discuss our current research and where it's leading. And I hope to update you soon on the next steps of this project and even potential outcomes. Thank you.